I'm sure you already know that the sum of what an organism is, its physical characteristics, metabolism, reproduction, and evolution, are all encoded in its genes. The same is true for microorganisms. In this chapter, we'll spend a little time comparing prokaryotic genetics to eukaryotic genetics, and then we'll talk about how prokaryotic genetics are different from those of the eukaryotes. That said, I won't spend a lot of time talking about DNA synthesis in eukaryotes or protein translation, as I assume you've already learned that in another class. If you need a refresher on what happens during those processes, I'll put a few videos in the lecture videos folder to help jog your memory. The objectives for this video are to define the terms genetics, gene, chromosome, genome, and nucleotide, and describe how they're related to each other. You should also be able to describe the central dogma of biology, and you should be able to compare the eukaryotic and prokaryotic genetics. I will be presenting a table that compares characteristics of the eukaryotic genome versus the prokaryotic genome. If I were you, I would learn that table. Hint, hint. Genetics is the study of what genes are, how they carry information, how that information is later expressed in an organism, and then how genes get replicated during DNA synthesis. A gene is a segment of DNA that encodes some sort of functional product. Now, usually it's a protein, but it doesn't have to be a protein. It could be an RNA, like a transfer RNA or a ribosomal RNA. Genes are carried on chromosomes. Chromosomes are structures that are made up of deoxyribonucleic acid, or DNA. And it's the DNA of an organism that physically carries the hereditary information. So if you look at this figure here, you can see what I mean. This end of the molecule shows you the DNA. You'll know that DNA is actually a double-stranded helix, so we can see the helical form here. And as we learned in Chapter 4, this DNA gets wrapped around histones in the case of eukaryotes, and then that forms chromatin. Then the chromatin fibers get twisted and coiled to form chromosome like this one. Quick question. Is this a chromosome for a eukaryote or a prokaryote? I knew you'd say eukaryote, and you'd be right. This is a linear piece of DNA, so we know that this is from a eukaryote. Another clue is that it's paired, and as you can see in the figure here, it's inside of a nucleus. So the genome of an organism includes all of the genetic information within that cell, or the cells of that organism. Now, your genetic information is encoded by nucleotides. It's the sequence of nucleotides that distinguish one gene from another. When we talk about a nucleotide, we're talking about a unit like this, where we have a sugar group, and in the case of DNA, this is going to be called deoxyribose. Attached to the sugar, we have a phosphate group. And together the sugar and the phosphate alternate to form the backbone of DNA. Then also attached to the sugar we have a nitrogen-containing base, and there are actually 4 of these nitrogen-containing bases: adenine, thymine, cytosine, and guanine. And it's the order of the adenine, thymine, cytosine, and guanines that make a gene what it is. That's where the code for eventually a protein will come from. Now you'll probably remember that adenine always pairs with thymine, and guanine always pairs with cytosine in DNA. The pair of nucleotides is called a base pair. So when you hear me talk about base pairs, I'm talking about A and T bonded together or C and G bonded together. The genome of an organism makes up the genotype. The genotype basically is all of the genes within that organism. but the genes are expressed differently. So the actual expression of the genes is called the phenotype. For a number of years, scientists have known that DNA in a cell is the hereditary material. It holds all of the codes for making proteins, and proteins are generally the workhorses of the cell. They make up the cell structure. They are the enzymes that allow chemical reactions to happen. They are the motor proteins that transport molecules and organelles from one location to another. They are nutrient sources in the case of some organisms. They do almost everything. They're involved in cell signaling. These are incredibly important molecules. To get from DNA to protein, however, you have to do two processes. The first one is called transcription. Transcription copies a DNA sequence into a related molecule called RNA. RNA is the molecule of protein synthesis. RNA gets converted into protein via a process called translation. Sir Francis Crick called this the central dogma of biology, that DNA is the hereditary material. It gets converted or transcribed into RNA, and then RNA gets translated into protein. Now, DNA has a variety of different functions in the cell and uses. It's responsible for the diversity of phenotypes. What does that mean? It's responsible for the different ways that organisms appear and the different characteristics that they have. DNA also can be looked at to determine how related two organisms are. Now, as eukaryotes, you're probably familiar with this in terms of paternity tests or those like Ancestry DNA analysis kits. But we can do the same thing for microorganisms as well. If we sequence the DNA for a particular gene in two different organisms, if the two organisms are closely related, we expect to see a lot of the same A's, T's, C's, and G's in the same positions among those two genes. If the organisms are not very closely related, we might see that less than 30% of the A's, T's, C's, and G's are actually matching between the two organisms. Because DNA is so important to making an organism what it is, we can use genes in known organisms to identify unknown organisms. So let's say you have a patient that comes in, they have a bacterial infection, you suspect that it's Salmonella typhimurium. If you have a little bit of DNA from one of the genes of Salmonella typhimurium, you can use that to probe the DNA of bacteria in the patient's body. And if the patient actually has Salmonella typhimurium in them, then the DNA from your known organism should bond with DNA from the patient, allowing identification. And then, of course, in Chapter 1, we talked about how recombinant DNA technology and gene therapy and other biotechnology techniques allow us to manipulate the DNA in living organisms in order to produce useful products. Before we get too involved in the nitty-gritty of this chapter, I'd like to provide an overview of some of the differences between your prokaryotes and your eukaryotes in terms of genetics. One difference that you're already familiar with is that the chromosomes are different. Remember, prokaryotes have one circular chromosome, and eukaryotes have linear paired chromosomes. And this is almost universally true. About 10% of prokaryotes may actually have paired or linear chromosomes, but the vast majority of them have one circular chromosome. Because the bacterial chromosome is circular, that means that DNA synthesis happens a little bit differently for your prokaryotes than it does for your eukaryotes. One difference is that there's a specific site on the chromosome called the origin of replication, and the DNA nucleotides at this site are designed to bind a DNA synthesis initiator protein called DNA-A. So as more DNA-A binds at the origin of replication, it becomes easier and easier for another protein called helicase to actually open up the DNA, forming a bubble called the replication bubble. At the ends, opposite ends of the bubble, so here and here, we're going to have two Y-shaped structures called replication forks. And this is where the helicase, along with other replication proteins, are going to bind. Most importantly, DNA polymerase is going to bind there. As DNA polymerase reads the nucleotide sequences at the replication forks, the replication machines at the forks will begin to move in opposite directions. This makes the bubble bigger and bigger. That's the replication bubble, by the way. It gets bigger and bigger, and the replication forks get farther and farther away from each other. Replication happens in two directions, in two opposite directions. So we call this type of replication bidirectional. Replication continues in opposite directions until the two replication machines basically meet at a site that's opposite of the origin of replication. And this is where termination happens. So DNA synthesis stops. Through a process that we don't fully understand, but we know some of the enzymes involved, the two new circular DNAs will be separated from each other. And then each of these can go into two new cells. In eukaryotes, your chromosome is much longer than in prokaryotes. I believe I mentioned that your typical prokaryotic chromosome is only about 1 millimeter in length. For a eukaryote, your chromosome can be something like 6 feet long, so quite a bit longer. In any case, there will be multiple origins of replication along the eukaryotic chromosome, and replication bubbles can form at each of those origins of replication. Then, kind of like what we saw with the prokaryotes, replication proceeds in opposite directions along the chromosome until eventually our replication bubbles meet each other and have to fuse together, producing two daughter strands of DNA. So it's slightly different. A couple of other differences that we see between prokaryotic DNA replication and eukaryotic DNA replication is where replication happens. In a prokaryote, it's going to happen in the nucleoid, but in a eukaryote, it's going to happen in the nucleus. For your prokaryotes, typically there's only one origin of replication, but for your eukaryotic chromosomes, there can be over thousands. We'll get one replication bubble for your prokaryotes. For your eukaryotes, we'll get many replication bubbles. This also means that we get two replication forks for our prokaryotes, but many replication forks for our eukaryotes. Finally, prokaryotic replication is a bit faster than it is for your eukaryotes. So prokaryotes, about 2,000 base pairs per second get added to the DNA. In eukaryotes, it's only about 100 nucleotides per second added. Why is it so slow? Well, it has to do with the DNA polymerases. For your eukaryotes, DNA polymerase has what's called proofreading function. So once the polymerase inserts a base opposite of a nucleotide from our template DNA, then the enzyme checks to make sure that that's the right base. And if it's the wrong base, the enzyme stalls until the base can be fixed. Now let's talk about a few other differences. So, as you're already familiar, the way that your prokaryotic and eukaryotic chromosome is compacted is different. Remember, in your prokaryotes, your chromosomes are bound by DNA binding proteins that loop the prokaryotic chromosome, and then enzymes like gyrase and topoisomerase allow supercoiling to happen. This is different from eukaryotes that wrap their DNA around histones, forming chromatin, and then the chromatin gets twisted and coiled and compacted. We also talked about how transcription and translation happen at the same time in prokaryotes because the DNA for these cells is in the cytoplasm, where ribosomes will be floating around, as well as the proteins involved in transcription. In eukaryotes, remember, transcription happens only in the nucleus, where the DNA for the cell is located. Then the messenger RNAs diffuse out of the nucleus, go into the cytoplasm where ribosomes are, and then the ribosomes do translation. So it's sequential. We also talked about how there's only one circular chromosome for your prokaryote, so that means that most of those organisms are haploid. Eukaryotes have paired chromosomes. They have two copies of the same chromosome, one coming from mom, one coming from dad, and so this means that these organisms are diploid. Prokaryotes may have plasmids. Remember, plasmids are these small extrachromosomal DNAs. They can have less than 30 genes on them, and the genes are typically non-essential. Eukaryotes, with the exception of a few fungi, yeasts primarily, don't have plasmids, so that's almost exclusively something we see in prokaryotes. Prokaryotes have operons. So if you think about an organism, a living organism, they have a lot of complex processes that happen. And these organisms will then require coordinated functions from many genes. One solution to this is to organize the genes into functional cassettes called operons. And we'll talk about those in a bit more detail in another video. Operons are extremely rare in eukaryotes. In fact, so far, I believe we only have seen operons in, in fruit flies and nematodes. Everybody else has some other organization of their genes. So it's almost unique to prokaryotes. One other difference that I want to mention before I wrap this video up is that prokaryotic genomes are efficient and they have very little repetitive DNA. So repetitive DNA is going to be a DNA that has the same nucleotides repeated over and over and over times. It could be repeated a handful of times, like 3 times, or several hundred times. So you might see TAG, TAG, TAG. Okay, and eukaryotic genomes have lots of these. In fact, eukaryotic genomes have so much repetitive and sometimes it's called junk DNA that really only 2% of your DNA makes you human, and less than 1% of your DNA makes you the unique person that you are. Prokaryotes don't have a lot of repetitive sequences. They don't have a lot of junk DNA. In fact, it's less than 12%, so much more efficient genome than for your eukaryotes. So that's it for this video. We talked about what genetics are. Remember, we said that genetics is the study of genes and their function, how they're replicated, and how they're expressed. A gene is simply a segment of DNA, and it's the genes in an organism that determine the organism's structure as well as functions. Genes are housed on chromosomes, which can be circular or linear strands of DNA, deoxyribonucleic acid, and genes are composed of nucleotides, the A's, T's, C's, and G's. Remember, it's the order of the A's, T's, C's, and G's that makes or distinguishes one gene from another. And as we'll find out later in this chapter, it's that order that also determines the sequence of proteins. All of the genes in a living organism make up its genome. So since we're talking about um, microorganisms, a lot of times the microorganisms are unicellular. That means that all of the genes for that organism are located in one cell. We also talked about the central dogma of biology. Remember, this was a term that was coined by Sir Francis Crick that relates DNA to protein and says that DNA is the hereditary material for living organisms. All of our genes are encoded in our DNA. Proteins are the workhorses of the cell. They have lots of functions, and I believe we named some functions for proteins. The way we get from DNA to protein is that DNA has to be transcribed into RNA. RNA is the molecule of protein synthesis. Ribosomes can bind the RNA, in this case it's a messenger RNA. It binds the messenger RNA, and with the help of two other types of RNAs, a transfer RNA and a ribosomal RNA, the ribosome converts the sequence of nucleotides into the language of amino acids, aka a protein. So that's your central dogma. And finally, we went through a table that compares a few select facts about prokaryotic genetics to eukaryotic genetics. In the next video, we're going to start getting into the nitty-gritty. We'll start talking about operons and how they work and how they're regulated. So I'll see you at that video. Okay, here, let's just try 3 review questions. Which of the following is a characteristic of a prokaryote? Would that be paired linear chromosomes, 98% junk DNA, histones, or operons? Operons! Great job! Transcription is it DNA synthesis from an RNA template, RNA synthesis from a DNA template, or the process that makes protein from RNA? RNA synthesis from a DNA template. Right! RNA is basically a RNA copy of a DNA gene. Which of the following is true of prokaryotic DNA synthesis? Is it that it begins at the origin of replication, or that it's bidirectional, or that it's fast compared to eukaryotes? All of the above. Absolutely correct. Good job.